life's like out in the Sargasso Sea, and today I want to talk a little bit about some of the ecological perspectives within the Sargasso Sea and the community. I won't get sidetracked by lionfish and lobsters, although they're very important elements in Bermuda. What I want to try to understand, help you understand, is looking at the real extensive scales by which, when we think about the Sargasso Sea, you really have to keep that perspective, looking at the very vastness of the system and remembering we've got both spatial and temporal dimensions that are very important when you look at some of the biology of the animals, to some extent the plants that we have here. So if you think about this large basin of the western, eastern Atlantic, they're connected together. The Sargasso Sea is an element that knits those communities together. But that's several thousand kilometers in spatial scale. And we have to imagine that many animals today, we're learning, I'll give you some examples later on, are capable of exploiting the resources in the ocean at that scale, much beyond our imagination until we were able to track them in real time. And recognize that this plant is circulating throughout the Sargasso, even though it's not, an individual clump may not live that long, it's going to fragment, that's its only method of reproduction. So at a genetic level, there should be some level of homogeneity in there, although I hope to read a poster today that's going to tell me something different about that. But there's a largeness here which is very difficult to comprehend and very difficult to work at. And then you can shift from that large space scale down to this individual level of a clump where so much life is packed into just one lump of this weed. There's so many different invertebrates and microscopic plants attached to it. Many of these animals are going to be just stuck on that one plant for its entire life cycle. It's not going to go anywhere. So there's this difference in scales. It's quite remarkable. We also have to think about the temporal scale of the animals that are in the Sargasso Sea and recognizing that some animals are going to be utilizing the resources of the Sargasso Sea for some part of its life cycle and maybe over many decades of its life, if you look at long-lived animals such as cetaceans and sharks. And that this activity is also going to be determined in the vertical element in terms of the mass migration that happens every night as animals migrate up from the interior of the ocean to the surface waters to take advantage of feeding resources there. So it's really important to keep in mind that as we try to understand this system, it's at a scale which is very difficult for us to gather enough information to really truly understand all the nature of dimensions that exist for the animals that are using its resources. So the simplest question is, where is this Sargasso plant that so many animals become intimately associated by living on it for its whole life or being attracted to Sargasso to find food resources as it progresses through various stages of its life. So we look at maps of where Sargasso is, and this is still not showing you very much of the entire Sargasso Sea. We look at early distributions mapped by Parr and look at the data from the early phase of exploration by the S.E.A, Alan Stoner's work, to slightly expand the scope of measurement here. These come from the illustrations from Morrison Butler's paper. And we can go forward a little bit more in time and look at the intense nature of the S.E.A data set that plots out Sargasso in different seasons. But the maps are still limited. We really don't know much more about what Sargasso is doing as we go all the way out here into the eastern Atlantic. So we have a pretty good picture. Thanks very much for the great effort that S.E.A has done over time to tell us a lot more about the distribution here, but we still very limited in understanding of what Sargasso is and where it is, and therefore what other animals might be using it, because we just don't have that information. This may change going forward as we begin to use satellite oceanography to try to give us more information about the distribution. This is Bermuda tucked down here. Scale is not evident here, but there are algorithms that graduate students and faculty are using to try to determine what we can learn about the distribution of this very distinctive, highly pigmented plant in the ocean, but it's aggregating and disaggregating over time, and so the signal's there for a period of time, but if your satellite's on the other side of the planet, you're not going to capture it. So can we gather a data set over time that's going to give us a better spatial map of these plants, and also integrating that with the physical oceanographic and climatological features which occur to just sort of determine whether this material is put into long windrows or aggregated into large mats. And those characteristic patterns of spatial aggregation or disaggregation really are very important for how animals that are looking for something in the Sargasso community are going to find it and detect it and utilize it in some way. So I just want to go back to this idea of animals and their use of the Sargasso community in different ways, and sort of understand that we have animals that will cover vast distances, migratory animals, seabirds, sharks, whales, billfish, they're going to traverse thousands of kilometers in relatively short periods of time to find resources that they need there. We understand a fair bit about these mass migrations of eels coming from both Europe and North America into the Sargasso Sea, particularly to spawn and release their leptocephali larvae that will drift around and eventually make their way back to hopefully healthy rivers where they can continue their life as eels. They're a very highly valuable resource, particularly in Europe. I'm not diminishing the North Americans' interest in eels, but the European community is very concerned about it, and they're very interested. They spent quite a bit of money trying to study more about what are these eels doing, what do they need, where are they going to go, in order to hopefully have a resource that they can use back in their nation states. We look at migratory seabirds that are moving in both within the northern hemisphere, but also animals that migrate up from the southern hemisphere to use the Sargasso Sea. There's a long list of animals. We have two particular species, the longtail and also the endemic Bermuda cahao storm petrel, that breed in Bermuda. The cahao only breeds in Bermuda. The longtail also breeds in the Caribbean, but they migrate in on a seasonal basis to carry out their breeding activity. The longtails are just starting right now. The cahaos are finished. The longtails will, from what we understand, spend a fair bit of time up feeding around Bermuda. The cahao doesn't seem to do that. While it's breeding in Bermuda, it's actually making on a nightly journey of several hundred kilometers north to the Gulf Stream to actually forage, because there's a bigger payoff for those birds foraging, where it's more highly aggregated animals for it to feed on. There's a lot more I don't know about seabirds. I was on a cruise recently on the Sea Dragon. We found 12 species of seabirds when we were out there for just 6 days. I was really familiar with only 3 of those, the greater shearwater, pictured above, and also the tropic bird. This was only the 3rd record of the red-billed tropic bird. It's a more tropical species, tends not to come up how much we read, but we were out there and we saw one. So it's obviously using those resources in the Sargasso Sea south of Bermuda on a more regular basis, looking at the South Polar Skua, moving northwards to escape the winter of Antarctica at this time of year. So there's a lot more going on that we have to learn about these patterns of distribution of these migratory animals and understand what they're doing there. We can also look at pelagic fishes. I'll show you a graphic in a minute that looks at the migratory patterns of tunas. There's many other species we have not got very detailed information about. We have much to learn there. We know a fair bit about some cetaceans, mostly because the migratory humpback whales are passing Bermuda fairly consistently every winter. And we have a much larger program that's been growing over the last few years to identify the animals. We've taken our catalog from about 150 known animals back in 2005 to 880 animals that we identified just in the last 6 or 7 years. Still much more we have to uncover. And in general, we have to point out that all of our Atlantic sea turtles spend time in the Sargasso as juveniles before hopping off at about this size, either into Bermuda's coastal environment to take up, in the case of the green turtle, life in our seagrass beds, and also into anywhere in the Caribbean basin, as well as back into the Mediterranean. So many different species feed into the Sargasso Sea to be able to find resources at some stage in its life. But we have so much more to understand about patterns of movement, and especially the feeding relationships. So just to emphasize this point, this is some tag data of migratory tunas, the western breeding bluefin tuna and the eastern breeding bluefin tuna. And you can see for these dots, we're just over 1 year's data, that they are penetrating and using both of the populations, the resources of the Sargasso Sea. And here's just an insert of just one fish's data over a course of a year. Each color is just 1 month of data, so it's spending on average 2 months of its time just circling around Bermuda, finding some resources. I don't know what they're eating. I don't know what their diet consists of. I suspect large bluefin tuna are probably eating other smaller tunas. But just knowing that temporal pattern is important and understanding the spatial pattern for these animals across here means that the resources in the Sargasso Sea are relevant for that particular species, that particular time. And if we can gather more data and it accumulates, we get a bigger picture. We've recently collected tag data from tiger sharks. We tagged off Bermuda. They're showing pan-Atlantic patterns of movement on a temporal annual basis. We've been tagging our endemic seabird, the cahao. We find that when they leave Bermuda, they don't really spend any time in the Sargasso Sea. They move up to the west coast of Ireland, off the Azores. Just this summer, we're hoping to get back some data loggers from our longtails to find out just where they are foraging on a daily basis. On this cruise that I was on, we were 200 miles south of Bermuda, and there was ostensibly a Bermuda longtail. That was my assumption, because Bermuda is the nearest landmass. But these data loggers will give us that window of information, information about what these animals are actually doing out there. Other sources of information about the value of activity in the Sargasso Sea is derived from fishery statistics. If you're looking at fisheries data, this is the bottom of a two-panel graphic that I stole from a Bermudian master's student. Looking at fisheries information, they tell you there's stuff out there people find valuable. Looking at when the U.S. entered the yellowfin tuna in the Atlantic, and this is box 5, the latitude and longitude that we sort of consider the Sargasso Sea. Look at the levels of extraction, and everything sort of cuts off right here. If you looked at the top panel that shows you the catch per unit effort, it does this. So as they keep investing more fishing gear, fishing effort, the returns weren't there. I'm sure there are lots of yellowfin tuna out there. We catch them regularly in Bermuda, and our goal in the long run, of course, is to have a healthy ocean where we can continue to catch fish at some sustainable level. But you know, for this particular species, several other large commercially extracted species, they show us that the Sargasso Sea has been and hopefully will have a future as a place for large pelagic fishes to be successful in moving around in there. But that's just catching them, not really knowing much about their biology, but I'll get back to that in a minute. Now we can go from that large scale of large migratory animals moving around down to this intimate scale of life within the Sargasso community, many hundreds of species, pretty well described, pretty well understood from the collections made here, made by scientists earlier in the 20th century to know who's living in there. We understand that there are animals that are attached here. They're primarily, in many cases, hydroids filter feeding, extracting planktonic nutrient resources, and to be useful as they excrete those nutrients into sustaining the growth of sargasso in a relatively oligotrophic situation we find in the surface waters of the Sargasso Sea. So we have some idea of what's living in here, but it really hasn't had a whole lot of attention. And some of the, something's not working here.
There's another, you're just trying to get to the next slide?
Trying to get to the next slide.
There's another key.
Okay. Technology.
Stand by.
Okay, so we, is that right one? Or there was one before that. Okay, there we go. We seem to be working. So from that aggregate information about the diversity of invertebrates primarily and fishes that are often associated with it, we can build a model of who eats who in the Sargasso Sea. A lot of this is derived by the work by Morris and Butler to look at the transfer of hydrocarbons, because this early study of the Sargasso Sea was really driven by the concern about hydrocarbons aggregating as tar lumps back in the 70s. We managed to solve that problem, but by tracing the hydrocarbons through the food web, we get some idea of who's eating who, as well as dietary analysis. So we have a model of what this community of life in there is, and all these animals in here, listed here, various shrimps and crabs, are going to be opportunities for fishes to recruit in there, while we have endemic fishes such as Histrio, the sargassum fish, and the sargassum pipefish who make us. Many other fishes come in there as larvae, begin to develop, using the sargassum as a nursery habitat is very critical for that. So we have a model of what's going on here, but we still have a lot of questions, and particularly since the effort to study the sargassum has really sort of diminished, we really don't know a lot about the sargassum communities in terms of, are they the same around Bermuda? Are they the same 700 kilometers to the southeast? Where do they lie closer to the Gulf Stream, north of Bermuda and south of Bermuda? We don't have a really good spatial map of what these communities look like. And particularly, we want to know, have they changed over time from the intense work done in the 70s, while S.E.A. has continued to accumulate a good library of information. We want to use that and begin to ask other questions. We look at calcifying organisms that make up a significant part of the sargassum community, our barnacles, our bryozoans in particular, any of the crustaceans. They're going to live in a more acidic ocean. What's their fate going to be living under these changed conditions? So we can look at the historic data that we have here, and a great bulk of it has come from S.E.A. in different ways. But we also have to remember that insight really comes from relatively limited spatial and temporal dimensions. We showed you the cruise tracks, which are nicely repeated because it's very good to come to Bermuda and then leave to and from Bermuda to get south or come north from there. But that's done over a relatively small period of time, over a month or two period. So we need to try to expand that. But even within that data set, we begin to see there are differences within the sargassum community at a spatial scale. We don't really know the temporal scale at all yet. And that's telling us that things are not uniform. They're not identical. The communities don't work that way. So in that, when you understand that variation in community structure may may exist, then you have to realize that that model we have about who's eating who may not always be applicable. So we can look at this information that comes out of Stoner and Greening's paper, looking at some of the distributions of stations occupied closer to the Gulf Stream, west of Bermuda, versus ones that are around Bermuda, slightly south or north of us. And here's an example of the top of that pyramid, the Sargasso swimming craft, or S.E.A, quite abundant in all the stations over here in the Gulf Stream, completely absent from all these stations. Look at this Cameron amphibod, Synanthothoe, very common in the Sargasso Sea stations, completely absent over here. So that lack of homogeneity means that you have to not fall into the assumption that all these systems are going to work exactly the same way. Whether you add or subtract different species over time, what are the outcomes going to be? Maybe this is just so limited because of the space and time in which it was sampled that on the broader scope of movement and pattern that really does all average out. But you wouldn't want to hold that assumption too rigidly until you had a chance to really look at it more closely. The other thing I wanted to bring to everybody's attention is to recognize that the Sargasso Sea is just at the surface, and there's all this life below it, which is very active and very dynamic, particularly with this nightly migration of zooplankton and all the things that are chasing the zooplankton up into the surface waters. And Bermuda's great advantage is that we're out in the middle of it, and from time to time, lots of stuff dies and washes up on our shore. So we keep finding examples of animals without having to do a whole lot of effort except to be aware of it. And living in Bermuda, most people are quite curious about things. So when a fisherman finds this gigantic deepwater squid that they haven't identified, and that eyeball is about that big, they get to bring it to me. They get to figure out, okay, here's a denizen in the deep. What's it doing? What's it been feeding on? Likewise with these other fishes that we have here. Remarkable diversity of life in the inner ocean that we still really don't have a very complete picture about because it's so difficult to get out there on a regular basis to really study and understand it. Other examples of flotsam and jetsam I find on the reef, this roughly up here, probably more associated with the seamount than as a pelagic animal. I don't know much about bromets. We get quite a few of these large homostrepes, squids, regularly stranding in Bermuda that are brought in to look at. So there's a lot more life in the interior of the ocean, and what are its connections to the surface waters in terms of some of the things that we are concerned about. Here's an example of another find. This was a set of Myctophis. These are lanternfishes that vertically migrate every day. I found these along one beach one morning, and these animals have migrated up to feed on particles. We're really concerned about particles of plastic in the ocean. What is a burden for these animals in terms of taking on those plastics? Haven't had time to dissect all these. Any volunteers want to come and dissect all these fish guts? They're not very big animals. So there's just much more going on in the Sargasso Sea than what's obvious when you fly into Bermuda or you're sailing along. You can see these rafts of seaweeds. There's the opportunities for life to use the resource in the Sargasso Sea are quite varied in dimension. It's important to keep that in mind. Just thinking about this life in the deep ocean, this is a graphic from Sutton that looks at the abundance of deep sea fishes that are going to be either in relatively shallow waters, the pelagic species versus deeper species. Here's Cyclothone browii. That's the champion in terms of its frequency of abundance in there. Again, probably a particle feeding predator. What do we know about the impact of, say, pelagic plastics or other contaminants on this particular fish species, or indeed the entire interior population of fishes? Much more to be learned there. So I just want to finish up now to talk a little about some of the efforts that we're doing in Bermuda with respect to conservation in the Sargasso Sea. And we are sort of involved in two sort of slightly intermeshed but slightly out of sync efforts. And one is the work with the Sargasso Sea Alliance, which is a privately funded group that has an office in London, led by Dr. David Freestone, who's an environmental lawyer. And the idea is to try to build a broad alliance to set aside parts of the Sargasso Sea as a marine protected area. And the track they're taking is really to try to engage intergovernmental cooperation through the offices and various agencies of the UN to be able to build up a sense of, well, we could work together to do this. But I think it'll fall into many of the cases that Tundi has highlighted before. Just saying that the place is the one thing you have to worry about is not going to be sufficient. And in many cases, it's the biology that should drive what that thing looks like. If you happen to know that all the tiger sharks are going to be spawning in Bermuda, well, you want to make that the place that's a core area that you have to look at. If you don't know much about what's going on out here because no one's actually been out there to sample, you should probably take some time to go and look for some interesting biology before you start drawing lines and making agreements with what you can or can't do in different places. So it'll be a longer term process, but we seem to be building some momentum. We've had some pretty positive interactions at the UN level to identify the Sargasso Sea as an area of special biological significance. That's a big step forward. Another effort a bit closer to home is working with the Pew Global Ocean Legacy Group to look at Bermuda's exclusive economic zone, which extends 200 nautical miles in all directions, that sort of inner circle around there, and try to look at setting aside some of that area as a marine protected area. And it's been getting a lot of press. We have an alliance of our NGOs to working towards trying to get the word out that there would be some value. And just about 2 weeks ago, we sat down for the first time between our commercial fishermen who spend time offshore, usually going after billfish or tuna. We have one long-line fisherman in Bermuda, the NGO groups, and the government. And it was our first meeting to try to figure out what's going on. It was a really interesting meeting because for the last 2 years, the NGO groups have been saying, we need protected areas, get out there and protect it. And the fishermen said, well, it already is protected. We've got an exclusive economic zone. Nobody can go in there and do anything. And the fishermen said, well, you know, if you draw a line in the sand and say, well, we'll protect all the area of water 100 miles to 200 miles, which would be 76% of Bermuda's waters, that would be a big number to protect. But the fisherman's response was, but I'm out there fishing, and I'm following a piece of satellite oceanography telling me this is the temperature front where I'm most likely to find my fish, and I'm at mile 95, and you're telling me I can't go to mile 100 to catch that fish for the 2 days I'm going to be out here? I don't know. There's a lot of interesting ways to take enthusiasm for protection, but make it work at the level that's going to allow for sustainable extraction. So we hope to move forward with that in a positive way, but it's slow going, but I'm optimistic we'll get there. We do a fair bit of work stimulated by the arrival of the Sea Dragon and the Five Gyres group to look at marine debris, marine plastic debris specifically. This is a picture of our very first sample, 248 grams of plastic junk. That was just along 25 meters of the high tide on Bermuda, and this is some of our data since that time. There's quite a lot of variance depending on beach. Some beaches are better traps at collecting plastic debris, others are around 5 or 10, but it's never zero. Every student group I take out there, there's always something. You can say, oh, where's this piece of plastic? And that gets their attention in a hurry. We hope to continue to work with the Sea Dragon and especially S.E.A. to think about more about plastic. The plastic data that S.E.A. has collected over the last few years is really quite remarkable. Last few decades, I should say, not years. It really provides a great context for what we're doing. We'd like to use Sea Dragon, if we can raise the funds, to go further east of Bermuda, that area that doesn't get much attention, to see what we can find out there. And we have some partners that are trying to continue to work on that. So I'd just like to conclude by saying that we know a lot about the Sargasso Sea in many ways. We know a lot about certain species, but there's a lot of other species that we really have much more work to do. And I think we have to get the biology straight if we're going to be effective in trying to come up with the best conservation strategies. It's no good saying we're going to protect something, there's nothing going on there because you lack the information to know where significant biological events occur in terms of migration, reproductive activity. And I hope that we can be effective in our conservation efforts to try to work with any partners to try to do a better job of keeping some of the threats down. But the threats aren't going to go away. The plastic is just, it's going to be a long-term problem. Thank you very much.
Thanks so much for the invitation. And it is my pleasure, I get to tell you a little bit about some of the fun things that we do with what I consider, and I feel like I'm among a friendly audience because I'm going to present a fairly complicated problem. And I'm surprised I haven't heard the term, but now that we're in New England, I feel like we should be calling it what it is, a wicked problem. Some of these wicked problems are, these multifaceted, very complicated questions out there that really are a burden to solve. And so I'm a rather odd person in the field of toxicology because we're trying to deal with questions in animals that it's almost impossible to really address the question the way you want to, because you can't take those animals and put them in tanks and dose them and have any sort of control in your experiments. And so we go about this problem by trying to look at many different things together and try to piece it together and sort of follow a weight of evidence approach to try to see what the problem is, characterize it as best we can, and then use that hopefully, and I'm not on the manager's side, but I like to think that the work will eventually influence management and policy. So I'm going to talk about integrating some that are some questions that are all complex in and of themselves: the chemistry, biomarker, and disease screening data. So first, show this picture for many reasons. So where in the world is Hawaii Pacific University? Well, I happen to be a fan of Google Earth, just like one of the students that presented this morning. And I love this picture because it does show just how remote Hawaii is. It is arguably the most remote place on the Earth. It is surrounded by other islands, but they're rather small, and certainly to the continents, it is the most remote island chain in the world. And so here we are. Oahu is right in the middle of it. And so when you think about, okay, I think that we existed for many years amongst the potential problem of contaminants in whales and dolphins. And Hawaii was kind of off the radar because where in the world is contamination going to come from? So you probably have a sense that most of the contamination, if our animals are actually exposed to some of these things that result from industry, is probably not from Hawaii, right? And so now that you've got a little bit of background in oceanography, you probably know that the Great Ocean Conveyor is going to be ultimately pushing water from the global oceans up this way to the North Atlantic. So you've got sort of a very broad swath of old water that could be carrying stuff. You've got the jet stream that is going to be basically primarily taking all air and pushing it this way, possibly raining things down over the ocean. So pretty much the signature you would expect to be all Asia and dropping over Hawaii and its residents. And then of course these are volcanic islands, right? And so we actually have a natural source of things that can be considered hazardous, like metals. We've got trace elements and metals that are actually being spewing up from the mantle for the last 30 years, by the way, with the longest active volcano. And then, of course, we do have Hawaii itself, which has a very long tradition of sugarcane and pineapple, which is dwindling and actually being replaced by a lot of organic farming and everything. But it does have a history of use of pesticides and herbicides. So there's lots of different places where the animals that are living here could, in fact, be exposed to contaminants. And then just to actually give you a little bit of a better sense of where HPU is, we're on the windward side of Oahu. And so we have a residence in the mountains of the windward side, as well as the far east. The arrow there is showing that we're on the far east side, and so that's where most of the marine science that I'm talking about today is actually happening. Why would we study Hawaii in the first place? Well, touched on some of these things, but some of the even more important things are, besides the fact that everybody wants to go there, there's a long and important history of monitoring in Hawaii. And so that includes the Ocean HOTS program that's been monitoring ocean acidification, the Mauna Loa CO2 monitoring that's been going on since the 60s, the Keeling curve that actually is really important in every single climate change talk that you come across. We can think about the fact that we have both resident and migrating species. So we have some species that are migrating very great distances, like the humpback whales, and we also have some that really will never leave one little part of their shoreline, which is a very interesting combination. And sometimes those resident and migrant are actually the same species. So you've got migrating pilot whales and you've got resident pilot whales. We have lots of endemic and endangered species in Hawaii in general. So endemic means that you are native and you're not found anywhere else. And so being a unique island chain, we have a lot of interesting species that may well be worth conserving and looking at and examining. And then, unfortunately, many of those are endangered as well. So they actually either have or deserve special protections. And those include marine mammals and seabirds. And of course, other rare species that make up the interesting dynamic that is Hawaii. So here's another picture that I like, because I like to point out that we don't have a very good sense of why the marine organisms, or at least most people, this group may be a little different, but in general, people don't think about what the ocean floor looks like. And so Mauna Loa is actually the largest mountain on the planet. You might think it's Everest, but it's not. Because when you add the 4,000 meters that come from the ocean floor, plus the 14,000 that are above land, you've got the highest peak on the planet. And so in those giant mountains in the middle of the Pacific, you have all these valleys and crevices and a really dynamic structure underneath of seamounts and nice steep slopes. And so we have a lot of very interesting surfaces for midwater fish and all sorts of benthic organisms to exist. And so Hawaii is going to, in the middle of this big oligotrophic water, we're going to have a lot of interesting places for food to grow, and so we're going to have a lot of attraction for all these marine mammals to come to Hawaii. Okay, so now I want to say that I will come clean and tell you that one of my least favorite questions from somebody who is asking about my research is, why did the animal die? So I run with a colleague of mine, Christy West, the Marine Mammal Stranding Program in Hawaii. And so we respond to dead marine mammals across the main Hawaiian Islands. Whenever an animal dies, we go and respond to it, and we try to get as much biological information out of that animal as possible. It's a long-term commitment to try to get any data out of any one species, because we have 22 species that are very common in Hawaii. So usually when we get a report and it's this species, 80% of the time it's wrong, because most people don't know most of the species that we have in Hawaii. So the why did it die question is one of those that I have to take a big breath and I have to kind of step away from my main thing, which is, I guess as a researcher, I have evolved to really cling to this idea of multiple stressors. And so I think of this as an oversimplification, but if you think of an organism, and you can even think of yourself as having a tolerance that is some amount. We cannot really define tolerance, but I will show it to you anyway. So imagine you've got your cetacean, and so let's just imagine that one of the things that happens to you is that you're exposed to some contaminants in utero, which might affect your brain development, and so your cognition, maybe your IQ, a little bit lower than it should be. Then imagine that ocean acidification actually shifts your food web, so your favorite food is just a little less plentiful. And then imagine that you're exposed to other contaminants, and I'm saying contaminants in general, there's many, many classes of contaminants, and they all, of course, can interact with each other and become a little bit better or a little bit worse. But we know that many of them are actually immunosuppressive, and so you would have a less of an ability to respond to a disease should you happen to be exposed to it. And then imagine that somebody decides to lay a new cable in Hawaii, and so dredging all of a sudden disrupted your favorite habitat. And then say it's a RIMPAC year, and so that there are major naval operations going on in the waters where your favorite food source is, and all of a sudden you don't really know where to go to eat, or you were actually blasted with some sound that forced you to surface prematurely. And then say you actually had a bit of an infection. You had a little cold. So all these things are, piling up on you and taking away from your tolerance. And then say sharks are in the area. And so you ended up succumbing, you dolphin, you ended up being vulnerable to a shark attack. Well, any reasonable person would say, of course, that animal died of a shark attack. Because by gosh, we've had a lot of shark attacks in Hawaii lately, and so actually I do suspect that we're not getting some strandings because I think that sharks are taking them away, so they don't even get a chance to see them. But so this would be the obvious response: is yes, that shark had caused the death of that animal. But I'm arguing that all these other things they take their toll. And so we may not be able to stop a shark attack, and it may not be a good idea to go out there and call all the Galapagos sharks that are moving in, or we don't want to take away all the tiger sharks either. So if we're not going to be prepared to do that, can we actually lessen the effect of these other things and make a positive impact? So that's why it's a wicked problem. That's a complicated situation. So The general approach that we use in general to assessing impacts of contaminants in marine mammals is to first look and see if it's exposed, then look and see if there's a physiological response, because just because they're exposed doesn't mean that it's going to cause a negative effect, and then try to link that to some sort of disease process. Now this is sort of, there's definitely a stochastic effect between this, and maybe even between the first and the second, but we try to draw the links in the whole process, while admitting that contaminant exposure to disease there's a lot that's not understood and a lot of randomness to that. And so, just because you smoke one cigarette doesn't mean you're going to get lung cancer. But there's plenty of variables in there that would suggest that those things are linked. So we have to accept that there's some things that we just won't be able to define. So how do we measure these things? Well, first, of course, contaminant exposure, we're going to actually look at the tissues of the animals and measure the actual contaminants. For a physiological response, we're going to look at a biomarker. So a biological enzyme or some other indicator compound in the body that will actually tell us whether or not a contaminant actually caused that response. And so to get there, we're going to have to rely on some other folks' research. We're going to have to look at mammalian models like mouse, or some other molecular biological tools, because it's going to be really hard to figure this out de novo in a marine mammal. And then for disease, we have to diagnose or confirm that the disease actually happened. So to some extent, my lab is involved in all of these things and at various levels of sophistication, depending on what we're talking about at the moment. So I'll first talk about, I will try to break it down and talk about one section of the research, and we'll look first at contaminant exposure. And so persistent organic pollutants is what I definitely spend most of my effort looking at. We also do some work on trace elements and metals, but this definitely covers most of what's going on in the lab. So we break down persistent organics into legacy and emerging, and those are not very good categories, because legacy means it's been around a long time, and emerging is what we called them 10 years ago or 20 years ago, and they may still be around. So they're no longer emerging, but we still call them that because they were a little bit more recently introduced. But your legacies include PCBs and DDT and dioxins, some of the more classic nasty compounds, and I was glad to see that somebody already discussed PCBs a bit, because those will come up again. And then emerging are some of the newer problems that are less well characterized, like PFCs, which include Scotch-Guard, Teflon, they're perfluorinated compounds. And PBDEs, flame retardants, and that gives the new car smell, and some of those, I bought a pillow that a nice neck pillow to come here, and I could just smell the PBDEs that are in there. And I just thought, oh, why did I do it? I knew that microfiber was going to have flame retardants. I'm sitting there trying to sleep when the EPA says it's okay. The EPA says it's okay. So HCBDs are what Europe calls flame retardants because they outlawed the PBDEs. And the phthalates are all over the place, and plasticizers and makeup, lotion, and so we expose ourselves to all kinds of interesting persistent organic pollutants, sometimes very willingly. So POPs have industrial and agricultural sources. They tend to be persistent. They bioaccumulate. You know all this, as I now know from previous talks. And then they have variable degrees of toxicity. And so some are particularly toxic by being endocrine disruptors, by causing developmental toxicity in utero, by causing cancer, immunosuppression, etc. And so this is just a bit of the paradigm is that, and this is very surprisingly reliable, but it partitions to the lipid fraction, and then by going up the food chain, it tends to be about 10-fold higher with every single trophic level that you go up. And so for something like a killer whale that is actually feeding on marine mammals, some of them eat fish, some of them eat marine mammals, the ones that do eat marine mammals have the highest levels of persistent organics that are the ones that are measured. So how do we do this? So here's the POP analysis. It basically follows a pretty simple and classic procedure that most people don't want to do because it's very burdensome. But this is what a student of mine did. Takes a nice clean section, this is a piece of blubber over there, cuts the skin off, cuts the edges off, and then extracts it. That's Melanie doing her ACE extraction. Then you do some size exclusion chromatography, then you clean up that sample, and then ultimately you run it through GCMS and LCMS. And so she looked at about 150 compounds to actually through this process, and then came up with, clustered those compounds into groups, and essentially, I have to show this. It's a terrible figure to show a general audience, but it just represents so much work that I have to give credit. But you can see that in our Hawaiian marine mammals, these are the legacy compounds that have been outlawed for 40 years. DDTs and PCBs still are at the top of the list of those being persistent in our animals, and it's just rather shocking. And so you see the difference between the mean and the median. And so that suggests that you have a few animals that are just off the scales, right? That's what that means, because the mean is so much higher than the median. And to make this data a little bit more accessible, this is not entirely unexpected, but if we kind of group all the dolphins, and like I said, we've got many, many species of dolphins. They tend to be the highest. So like we're just breaking out the DDTs and the PCBs, the nasty legacies. Oceanic dolphins tend to have the highest concentration overall. And then beaked whales, which you may or may not be familiar with, they tend to be pretty loner and pretty far offshore a lot of times. And then sperm whales, that's clustering the big sperm whale that everyone knows, Moby Dick style. And then the kogias, which are actually, East Coast are fairly familiar with kogia, the pygmy sperm whales and the dwarf sperm whales. Those are very deep divers, and they tend to eat not the fish that are right close to shore. And then baleen whales, of course, are not eating large fish, and so our baleen whales are comprised of humpbacks. And actually, they're also not eating in Hawaii. So we're actually sampling. This is a nice sampling of Alaskan humpbacks. We can think about it. But baleen whales, it fits the paradigm that you eat lower in the food chain, your contaminant levels are going to be lower. So there's been a lot of effort over the years to try to put this into some sort of perspective. But okay, what does this mean? What does all this contaminants? What do these numbers mean? And so there was a nice study that tried to summarize all the information that we had on marine mammals. And by this, we're clustering some seal data too. So going across some generally not supposed to cross boundaries, but if you look at where this study, this is actually somebody else's work, plotting out where their animals fell on immunosuppression, low sex hormone, impaired reproduction, etc. And then there was another study that actually defined, oops, I thought I had this in the right order. So our animals are popping up on this chart. So our animals we've got a bunch in the immunosuppression range. So those are the spinner elephants. And then we've got some of the killer whales, which includes killer whales and false killer whales and melon-headed whales that are in the possibly impaired reproduction range, but this is based on seals, and so it's not, we're going with whatever we could in marine mammals here. This is a different threshold level that another paper suggested that might be a 2002 paper. So that just kind of gives you an idea that we are, in Hawaii, I don't know how you feel about whether or not, I should have forced you to commit in your own mind about where you thought maybe our animals would be. Whether it's pristine paradise or whether it's pretty much, a soup of contaminants like everywhere else in the world. That's a lot closer to industrial sources. But here we are, spanning the gamut of contaminant levels, and we're in the middle of nowhere. And so the DDT story is a bit similar. And again, a big range is the log scale, so it's not that big of a range, but some rather low levels for total DDTs. And then we've got some higher levels that definitely cross the boundaries of, the possibility for immunosuppression and tumors and so forth that have been established in other species. And so it's basically saying that all these things, that we're not the worst of the worst, but we're, it's certainly a possibility. The other thing that's not implicated in here is that for total DDT, been outlawed for 40 years in the United States, we have a much higher, I shouldn't say much, but we have a higher ratio of the parent compound than the degradation product. So there's DDT will eventually break down into some other products, DDE, and so we actually have a higher amount of the parent compound, which suggests that we're seeing the signature from Asia, where it's actually not banned. And this is a good question to pose to this group because these are social compromises that are made. So DDT is actually the best thing that you use for malaria control. So you must control mosquitoes to control malaria, and so that's a choice that several areas have gone ahead and made, and yet this is it will show up in the environment because it is so persistent. And so next step after we establish that yes, in fact, we do have contaminants, what do we know about their effect on health in the Hawaiian animals? Not just putting it up on a scale comparing to other investigators' work. And so remember we're in the second stage of the analysis here. And so ever since I was a graduate student, I've been thinking about biomarkers. And so they come in at least 3 flavors. And so I like to think of it as susceptibility. So you can look at something that would actually give you some idea of your genetic risk. And that would be something, your risk. There's several diseases that you can think of that would indicate that you might, if you are exposed to a compound, you might have a better chance of actually succumbing in a negative way to that compound. So that's susceptibility, indicating your genetic risk. Then there's the actual exposure, or your actual environmental risk, based on where you live and what you're exposed to, and then effect, which is combining the 2. And so I'm daring to show this because you've got a lot of molecular biologists here that are not going to be afraid of a cell pathway. So, this is one really great general way to think about what a contaminant is going to do when it comes into your cell. So if you start up there with a PHH or pH, so a general persistent organic pollutant, it's going to just flow right through that bilipid membrane, right? So no active transport required. It's going to, certain ones of the nasty compounds will actually bind to a particular receptor called the aryl hydrocarbon receptor. My Kenyan college friend is painfully aware of the aryl hydrocarbon receptor, and its dimerization partner is in the nucleus. So that whole complex is going to go into the nucleus and find its dimerization partner, and then that will actually, it will land on a promoter region called the dioxin response development, and then you'll actually get gene induction. So that compound is actually tracking all through the cell and actually turning on a gene, as part of this complex. And so turning on the gene causes mRNA to come out here and be transcribed and translated into a protein. And so cytochrome P450 1A1 is one of the very classic toxicological biomarkers that we can use to indicate whether or not this animal, this cell, had exposure to this particular compound. So very, very specific. And so your response can be good. This whole system evolved for a reason, right? So sometimes we can actually have detoxification of the original compound, and that's a good thing. We can be happy about that. Unfortunately, it doesn't always work that way. And so, as part of that evolution, bioactivation is part of the game too. And so sometimes you actually make something more reactive in the hopes that another enzyme will come and detoxify that. And then there's other options that actually cause a compound that was actually caused or created after the industrial age began. So it never even existed in significant quantities until 1940, which, now that you're all experts in molecular ecology, that ain't enough time to adapt. So you're not going to get anywhere in terms of an evolutionary response. So it's basically, some of the compounds are causing more harm than good. And so therefore you get sometimes altered cell proliferation or altered cell differentiation, and the translation of those two things are cancer and birth defects. So, and then oxidative stress is another general problem. And then overall what we're talking about is disease. So in the minor cases, some good things can happen, but there's a lot of ways that the system can go wrong. So I tend to think of this biomarker as having the potential to cause harm and to be an indicator of something not so good happening rather than something good happening. So, but nonetheless, it's an indicator that there are contaminants in the system. And so here is a little bit of data. So we've got some male killer whale, and this is a bit washed out, but this is actually showing that there can be induction of, those are the dark circles. So this is student data showing that we can actually highlight the cells themselves that are producing this protein. And so that is happening across the animal. It's happening in the skin, all the way to the urinary bladder and everything in between. And so in general, here are the trends, and this is what you would expect mostly from fat-loving chemicals. You have increased level with trophic level. You have more in males, and females have the chance to offload to their offspring. We have an increase with age, and so that you actually have more accumulation as the animal accumulates or ages, and then body condition shows that you can actually mobilize your lipids when you're sick and you're not eating. Your body needs to get nutrition from somewhere, and so they tend to mobilize the lipids in their blubber to actually accommodate their nutrition needs. And so you actually see a lot of induction of the biomarker when you actually mobilize contaminants that would otherwise be sequestered. And so the last bit of it is the disease piece. And so this is where the links get really dicey, but in recent years we have turned off to actually start looking more closely at diseases in our Hawaiian cetaceans. And so we've detected Brucella, herpes, crypto, and toxoplasmosis. And those are all bad news things. But the worst news is morbillivirus. This is a disease that is the cetacean version of measles. Morbillivirus is actually the name for the whole classification of measles and distemper. And so this was originally detected in a Maui Longman's Beach whale that stranded in 2010. And since then, we've actually looked back in the archive, and we've actually found it going all the way back to 1998. And it is actually a novel strain. So in Hawaii, we have our own strain of morbillivirus. And so this is something that brings up all kinds of molecular ecology questions that could be pursued. And so this is just showing that it has a terrible history, and it's the reason why cetacean biologists quake in their boots when they're thinking about morbillivirus, because it's caused massive die-offs in novel or in naive populations. And you all know this approach, so I don't need to go into this to sequence it. And so in summary of all this work, we've got lots of students that have contributed to this in many different ways. And so we're looking at everything from the contaminants to the biomarkers to the disease. And that's just the summary of the morbillivirus. And I also just in the scope of doing some stranding work, we actually do find ourselves interested in marine debris issues. And so here's a pilot whale that actually managed to swallow about 15 pounds of gear. And it's not very clear how long it was in the animal's stomach, but it clearly was actually imposing and stopping all digestion. This is all fishing line, popsicle sticks, zip ties, all kinds of blue plastic from who knows where. So quite a mess. And so overall, we've got multiple stresses going on in our marine animals, and we're trying our best to characterize this so that the information can actually be readily available for people like you to run with these questions and perhaps come up with some answers. So many acknowledgments to all of my students and supporting entities that support this work. And I know that I've gone complicated time, but if there's any questions, I'd be happy to answer.